Hello, is it audible to everyone? Audible? Okay. So thanks for coming to class. So today we'll be talking about the real class starts today, because the previous class I talked about what are the techniques that we use in the neuroscience to study neurodevelopment. Maybe a little bit. Okay. So today we'll be talking about neural induction. So before going to the specific term neural induction, I'd like to talk about what is induction. So if you are changing one particular substance or from one state to other state under the influence of specific stimulus, then we call it we are inducing to change from one state to another state. So similar thing we cell does, like when there is patterning of different tissue, like you have formation of neural tissue, formation of connective tissue, other different kind of tissue. So you have induction of one particular state to another state. So similarly, like here, the neural induction refers to the change of like cells or tissue under the influence of specific stimulating specific factor. You are changing to into the nervous tissue, like nervous system. You are changing to nerve cell. That we call it a neural induction. So there are specific signals which have been identified from the past. I'll be talking about slowly into the neural induction. What are the signal that is required, during development to change from particular cell type to neuronal cell type. So we'll be go one by one during the course of this, my lecture. So it's a relatively small lecture. I have only 27 slides because the last lecture, last year, the people who has taken the course, they have merged some part of the technique into this neural induction section. So, but since I have gone over the, the previous lecture about long topic about the techniques, so this will be relatively small lecture today, but we'll go slowly so that you'll be able to understand. Not changing. So almost all animals have nervous tissue, except these are the different, different species. You start from very lower animal like protist or flagellates or sponges. They don't have any nervous system, specified nervous system, but it starts from like cnidarians, like you have the hydra, which is radial symmetry, or the bilateria, which you can divide in one, like if you like chop those animals in a particular plane, you can divide into equal half. That is called as bilateral symmetry. So you have nervous system in proteostomes like mollusks or annelids, echinoderms, vertebrates, arthropods. Almost all animals have a nervous system, except this specific three group that I have mentioned, like protist or flagellates or sponges. Even hydra has the, like cnidarians, they have also the nervous system. So I will start the lecture with gastrulation, different stages of the development during development of nervous system. It starts from the very early stage, the embryonic stage. So one step, I'll be talking all these steps, starting from the production of a sperm and fusion with the ovum. Then you have the formation of a zygote. Then zygote cleaves to embryo, like divides to form embryo, and all these things I'll be talking about slowly. So, but before going to details into the development of the nervous systems, I just want to highlight about gastrulation. And this is the important step where, cell movements happens, and you have the development of nervous system during the gastrulation phase of this, development. So it is the simple term. The Lewis defined, the stages of the life. Like according to him, it is not the birth or marriage or death, but the gastrulation. He give importance to the gastrulation, which is truly the most important time in your life, because there, this is the stage where your nervous system develops. A lot of specification happens during the gastrulation phase. So here, this is the schematic representation showing different stages of the development in human. So starts from the production of ovum from ovary or sperm. Then you have oocyte. Then the fusion of sperm and ovum that results into zygote. This is the fertilization step. Then the zygote develop into two-cell stage or four-cell stage, or more. You have this morula stage in the human, but animals, but you don't have this stage in the amphibians or reptiles. You don't have this stage, so you have this. So I have the nice video showing how development happens in the human. I'll be talking very soon. So now you have the formation of blastocyst. It takes around 3 weeks to develop into embryo. Then you have the birth. So the basically during gastrulation phase, you have the neural induction happens during this gastrulation stage. So I'll be highlighting it soon. Let us talk about starts from the primitive, during the evolution, you have the amphibians, and this has been well studied, well established in the developmental biology, how the embryogenesis happens in amphibians. So you have this is the egg, and you have two different pools. One is called animal pool, and another is called vegetal pools. So you have the accumulation of yolk in the vegetal pool, and you have animal pools. Mostly the sperm fuses and form the zygote, and then you have the division. The zygote divides into since there is a lot of yolks are present, mostly the animal pool it divides from mass of cells. You can see that the animal pool divides into mostly the division happens here, and after some time, it forms mass of cell called as blastula, and it is just mass ball of cells. And after some times, during the development, you have the formation of blastocells, which is shown here like a open space called as blastocells. And here a lot of things happens during this development of the blastocells. So you have during from blastocells developing to gastrula. This is called gastrulation phase, where I talked about the neural induction happens during the gastrulation phase. So you have specific specific cells present in this gastrulation stage called as involuting marginal zone. So these are actually present near the blastopores. There is a specific pore called as a blastopore, and those cells invaginates actually inside the inside this in the embryo to develop into different layer of tissue. Mostly it forms the mesoderm of this during development. So you have initiation of neurogenic region here, which forms neural ectoderm. Nervous system develops in the ectoderm, and you have invagination near the those cell invaginates into the into this in the gastrula to form the mesoderm, and also you have endoderm formation, so which is also represented here. You can see that it invaginates from to the blastos in the gastrula stage to form mesoderm. Then you have endoderm. Then you have initiation of the neural plate, and also you have during the invagination it also forms the gut, the primitive gut called as archaeonteron. So I'll be talking more about this stage. This is called neurula stage, where you have initiation or formation of the nervous system. So this is just to highlight how the gastrulation happens and how it look like the gastrula stage. So as I mentioned earlier, so you have this animal pool and you have the vegetal pool. You have a lot of accumulation of this yolk, which provides the nourishment to the developing embryo, and you don't have that much division happens here in this region, the vegetal pool. Most of these cell divide in the animal pool, and they migrate to the vegetal pools. So and you have this cavity-like structure present in the animal pool that is called as blastocells, and the cells which are generated in the in the animal pool called as blastomere. So this is the structure of the frog embryo, like in the gastrulation stage. So the vegetal pool actually that becomes the future gut. Any questions? Okay. So as I mentioned earlier, the neural induction happens during the gastrula stage, and this is one of the structures that I want to highlight. I want to I have highlighted that you have there is a specific group of cell present in this region near to the blastopore, which is called as involuting marginal zone. So cells are invaginated inwards to form the mesoderm. And neural induction, there are several hypotheses. Some people believe that neural induction happens because the mesoderm gives signal to the ectoderm to initiate neural tissue. So we'll be coming about those hypotheses in the coming slide. So just remember that the neural induction happens in the gastrulation stage, the initiation of the neural tissue, or the specification of the neural tissue, that starts in the gastrulation stage. This is something important to remember. So thus I'm coming to the human, starting from the from amphibians. So here, just I want to highlight specific thing, which layer. So you have three different germ layer. You have ectoderm, which form the nervous tissue, mostly nervous system, skin, and some internal organs, which are formed from the ectoderm layer of this embryo. In the mesoderm layer, you have formation of muscles, you have bone and blood, circulatory system, and immune systems. And endoderm, mostly the gut and associated organ like liver and pancreas, which are formed from this three germ layer, which is highlighted here. So this is something that you guys should remember. So the question will come the multiple choice. I think you can able to remember. You don't just just go over this slide. Don't have to specifically kind of mug off these things. So just go over the slide. You will come to know. So now this is again showing the same thing, like structure of a frog embryo, and how the cells migrate to form three distinct germ layer, as I mentioned, the ectoderm, endoderm, and mesoderm. So here you have the dorsal blastopore leaf. So you have these are the future neural plate or neural cells are differentiated from here, and in the so you have a kind of some, as I mentioned earlier, you have some region in there is a specific region in the embryo which is called as dorsal leaf. So there you have invagination into the embryo, and that form mesoderm. The same thing, just I want to highlight that mesoderm invaginates from this specific region called as dorsal blastopore leaf, form the mesoderm. Same thing, kind of repetition here. The initiation of three different layer like endoderm, ectoderm, and mesoderm, and also you have from the ectoderm you have formation of the neural plate. From there you have generation of spinal cord, brain, and this stage is called as neural stage. This is something I have highlighted here. Also same thing I have shown here. And the notochord, that is also part of the mesoderm. So when there is invagination during the embryogenesis, you have formation of mesoderm, and also you have formation of notochord. The notochord can also give signal to the neural plate to form specific neural tissue. So notochord here playing an important role for the initiation of or for formation of the specific neural tissue. This is a video just showing how this thing happens. Just I want to play. And this is, you can see the cell invaginate into the inside, and this is how the gastrulation and neurulation happens in the frog embryo. So the important point to remember is that the invagination happens during the dorsal at the dorsal blastopore leaf, and that form different structure. One of the important layer of the germ layer of the embryo development is the mesoderm, and also you have formation of a notochord. So by the action of mesoderm and notochord, they give signal to the ectoderm, and that initiates neural tissue, formation of neural tissue. So the mesoderm gives mesoderm and notochord, they give signal to the ectoderm to initiate, to form the neural tissue in the embryo. That is something I am going to discuss in the coming slides. So next slide. So before in the previous slides, I talked about how gastrulation and initiation of neural tissue happens in the amphibians, and here I'll be talking about how this thing happens in the avian embryo, mostly the chick embryo. So similar kind of thing happens like invagination at the dorsal blastopore leaf, formation of mesoderm notochord, and initiation of give signal to the ectoderm to form the nervous tissue. So similar thing happens also in the chicken embryo. You can see that there are different layer here. One is the epidermal layer, that is the that form the epiblast or epiblast. It is the part of the epiderm. Now you have in migration of the cells, and that forms another layer called as mesoderm. As I mentioned, in the amphibian embryo, you have invagination from the dorsal blastopore leaves. So similar kind, you have the invagination. That is called as those cells basically forms from this primitive streak. So you in the amphibians, you have a dorsal blastopore leaf. In the chick embryo, you have the primitive streak, or this is another structure called as a Hansen's node. That is like dorsal blastopore leaf in the amphibian embryo. So in avian species, it is called as primitive streak or Hansen's node. So there you have invagination and migration of cells, and that form the mesoderms. So this is how you have formation of different layer in the avian species like ectoderm, endoderm, and mesoderm. And this acts like dorsal blastopore leaf like in amphibians. So here, any questions? Yeah. That is for the amphibian, like frog development of frog embryo. Yeah. So you have invagination from the dorsal blastopore leaf, the formation of the mesoderm layer, and also the formation of a notochord. So that is how you have the development of the frog embryo. Yeah. And the previous slide that I talked about the development for the avian species like the chick embryo. So similar like in the amphibian, you have dorsal blastopore leaf. They are the invagination. So in the primitive streak in the chick embryo, you have the like similar kind of mechanism also happening in in the chick embryo. That is called as primitive streak. So it is homologous to dorsal blastopore leaf of amphibians. Yeah. So is the human embryogenesis kind of complicated? I'll just go over this video to show you. Maybe I'll I don't know how to copy that. Sorry. I'm sorry, I don't know how to copy that, but you will have nice video explaining how. Can anybody help me how to do this? Okay. Maybe I'll stop there. No. Okay, I'll do that. Oh, this is weird. Where is that? Okay. So let me see. Anybody show me the YouTube? Okay. Oh. If we can open this. Okay. Yeah. Yeah. With it. I have to stop sharing maybe? No? No, because you see how. Yeah. There. Mm-hmm.
Perhaps. Let's see. No. Oh, it's because. Okay, this makes sense. They're able to see it on Zoom, but the screen is like a different setup. I think it's this one. But I don't know. Yeah. A single blue. You wanna write up? I did.
Maybe we can. It's just a video to like.
I just haven't watched it.
Yeah, maybe you can watch it at home. Yeah. Sorry.
I think it's yeah.
I think this one.
Oh, the screen is on.
Screen pass.
Resume. Yeah, and change.
I can stop this one, like maybe.
Yeah, do it.
Yeah, screen sharing has stopped.
So you have to share it again. Yeah, yeah. Yeah.
So, yeah, sorry. If you go to this link, so human embryogenesis is kind of complicated. So there are different steps. So if you can visit this link, there are this clear video explaining how human what happens during human embryogenesis. So okay, we know that different tissue arises from different germ layer of during the embryonic development. Which tissue, like the mesoderm, from the mesoderm you have formation of the connective tissue, or the blood from the ectoderm you have formation of the neural tissue, from the endoderm you have formation of a different organ. So but what are the signal, how this thing happens? Is it happen randomly, or there are specific kind of signals that specifies, okay, you have formation of nervous tissue because of this signal, you have formation of a connective tissue because of these signals. So we'll go some of these studies which are done from the past. So he's the famous German scientist Hans Spearman with his outstanding student Mangold. So they have they have talked about specific signal that is required for the induction of neural tissue. Basically, they got Nobel Prize together in 1924. Though his student complained that this is my finding, I should get only the Nobel Prize. So if you go over the story, since he was the primary author, like the PI, so she discovered this mechanism. What are the important factors that is specifying, developing to specific tissue? Like what are the signals for the induction of a nervous tissue? So she, for the first time, find out that, and everything is there in the internet. You can see that she has complained that I have identified this, I should get only the Nobel Prize. But since she worked with his lab, they shared Nobel Prize at 1924 for describing the mechanism of induction. So this is the very specific experiment they have done to suggest that there are specific reason during the development of the embryo that defines specification of specific tissue, or defines the formation of axis, like anterior posterior axis of in the animal. So what they have done, this is the very famous experiment, and for this experiment they get Nobel Prize, the Spearman-Mangold experiment. What they have done is they have two different gastrula. One is donor gastrula, another is recipient gastrula. So in the donor gastrula, they have they have this colored this is the colored gastrula. So they excise this colored gastrula, the part of the colored gastrula from the dorsal blastopore lip, and these experiments are done in the amphibian embryo. So they have excised this region of the cells, and they have implanted it another gastrula in the dorsal in the posterior side. Like this is the anterior side, this is the posterior side. So this is a recipient gastrula. So already you have the primary dorsal blastopore lip already present, which is non-pigmented, and this is pigmented dorsal blastopore lip, which is excised from this donor blastula. So and they found that these blastula, they develop into, twin embryo. Like you can see that the development twin embryo, and they become non-pigment later stage. And this non-pigmented, because if you look at this, they have excised the pigmented, structure from the dorsal blastopore leaf of one gastrula, and they put it in another gastrula, which is already pigmented. So one will assume that it will develop a pigmented kind like of structure, but it did not develop into pigmented kind of structure. It developed into separate, like transparent structure or non-pigmented structure like this. This is because you have something present in the dorsal blastopore leaf that is actually specifying or developing into the embryo, and that they called as the organizer of this embryo. That is that later they named as Spearman Mangold organizer. So that actually organizes the nervous tissue, and that actually organizes the pattern of this anterior posterior axis of this animal. So, and this is from her thesis, like Mangold thesis. So already you have this priming organizer. So they see what she have done is she has taken this organizer from another gastrula, put it here, and they develop into twin embryo. So this is the reason where there are certain things present in the dorsal blastopore leaf that is initiating the entire axis formation, entire formation of the embryo. So what are those things? I'll be talking in the next slide, in the coming slides. So another useful experiment they have done. So they said that during different stage of the development. So you have formation of zygote, then you have formation of cell mass, then that cell mass develop into gastrula. So this is the pre-gastrula stage. Now that develops into gastrula. So there are specific structure present in the pre-gastrula called as animal cap. So if they isolate this animal cap from the pre-gastrula stage of the embryonic development, and they isolate and culture it develops into epidermis. Okay, and if that pre-gastrula develops into gastrula, and they isolate the animal cap and culture it, and they found that they form neural, tissue. So there is something happening. Some genes are expressed when this is pre-gastrula is transitioned to the gastrula stage, and that genes or that signals actually initiating or forming different tissue. So in the pre-gastrula stage, the animal cap develop into epidermis, but when that pre-gastrula transition to the gastrula phase, because of the expression of specific genes, it develops in neural tissue. So what are those signals? We'll come into, we'll know it soon. So any question from this section? Okay. Now, another experiment they have also done. Here, this is DBL, dorsal blastopore leaf. This is a developmental stage of the amphibian embryo. And this is the blastopore, and this is a blastula. And you have the animal cap. So when they, culture this animal cap in the intact animal, they just, remove this mesodermal layer, and it develops into epidermal tissue. The animal cap develops into epidermal tissue. But when the mesoderm is present along with the animal cap, it develops in neural tissue. That means the mesoderm is giving signal to this animal cap, some kind of signal giving to the animal cap that is actually differentiating to the neural tissue. So as I mentioned earlier, the notochord and mesoderm give signal to the ectoderm to form the neural tissue. So this is the experiment they have done, that there is something present in this mesoderm, something, some signal is there in the mesoderm that is initiating to form the neural tissue. So there are some more experiments they have done, and they finally found out what are the signal, and they found that the signal is the noggin, which induces neural differentiation in animal cap. So there are a series of experiments they have done. So here this is embryo, and they develop into, this is the stage of the embryo that develop into another stage of the embryo. So you have dorsal side and you have ventral side, and this is a normal embryo, and you have nervous system in the dorsal side, and this is the amphibian embryo. Okay, so when the UV treated that embryo, you have loss of, you have loss of this dorsal nerve cord or the nervous tissue. That is they called as ventralized embryo. So you can ventralize the embryo just by the UV treatment. And if that is so, if you, and they want to highlight, they want to identify what are the chemicals, or what is the signal that is actually differentiating to nervous tissue. So there's a series of experiments they have just done. So either you can ventralize the embryo with UV treatment, you have a loss of the nervous tissue, or you can treat the embryo with lithium, and you can also hyperdorsalize the embryo. You have loss of the ventral part, so you have more formation of the dorsal part. And in the latter they extracted this, what are the signal present in this, that is specifying the dorsal nervous tissue. They extract the mRNA from this embryo, and they implant that mRNA or treated that mRNA to this ventralized embryo. They found that they forms into normal embryo. Is it clear? So you can do in two ways. Either you extract mRNA, or you extract total RNA, make into cDNA, and you transfect that cDNA in a vector to the ventralized embryo. You can develop into normal embryo. And with a series of experiments, they found that what is the gene that is actually inducing neural differentiation, and they found that it is the noggin protein or noggin genes. So they pinpoint that this is the noggin cDNA or noggin mRNA, which is inducing the induction of, which is inducing formation of neural tissue. So they found, in latter they did that. They just isolate the protein, noggin protein, and treated this animal cap, and they found that the neural tissue or a neural tissue is generated by the treatment of noggin protein. So they have done by cloning this noggin gene and transfecting that and expressing that in the embryo. So all these experiments suggest that the secreted protein noggin that is able to induce dorsal neural tissue. Okay, and later other group of people also find that it is not only the noggin, and codin or follistatin have also the similar ability that can induce the neural tissue. So if I summarize the things, we can say that from the dorsal blastopore leaf, it develops into different layer. It develops into mesoderm. Then mesoderm gives signal to the ectoderm. So those signals, basically, the signal is noggin or codin or follistatin. Those signals specifically, forming the nervous tissue in the amphibian embryo. This is just some of these slides showing that in the lab they have the control, and when they EV treated the embryo, you can see that you don't have formation of this dorsal nervous tissue. But when they inject noggin mRNA or noggin cDNA, they found that it formed the dorsal dorsal nerve cord, and also you have formation of kind of perfect embryo, developed embryo. And later also they found that so they have identified this thing by RNA in situ hybridization. They found that it is the noggin mRNA, which is actually they found that the noggin mRNA in the dorsal blastopore leaf. So that is actually inducing formation of the nervous tissue. So any questions?
I just had a question on the UV. Like, what is it doing? Is it just like getting rid of all of the noggins? That's why it's becoming ventralized, or is it doing something else?
It is actually killing the nervous tissue. Most of these, so it is actually neutralizing the dorsal blastopore leaf. That there you have this noggin, so you have a loss of noggin because of the UV treatment, because you are killing the cells which produce noggin there.
So then you can't form the dorsal, like the nerves.
Yeah. So when you kill that dorsal blastopore leaf by UV treatment, you have loss of this ventral dorsal nervous tissue. But when you are treating the ventralized embryo with noggin mRNA or noggin protein, or noggin cDNA, you can able to restore the dorsal nervous tissue. So here different set of experiment later people have tried, and they found that what are the signal that is actually forming the nervous tissue, or what are the signal that is inhibiting the formation of the nervous tissue. And as I mentioned earlier, that noggin, codin, or follistatin, they are inducer of a nervous tissue, but there are also some signal which inhibit the formation of a nervous tissue, because in the body or in the system you have always the homeostasis. There should be a balance. So the BMP, the bone morphogenetic protein, is considered as inhibition of the they inhibit the formation of nervous tissue. So there are set of experiment people have done to kind of suggest that the BMP is a inhibitor of nervous tissue. So here when you have isolated the animal cap and these are dissociated, it forms people have seen that it forms the nervous tissue. But when the dissociated animal cap are treated with the BMP, it forms epidermis. So the BMP actually inhibiting the formation of, or somehow it is competitive, binding this receptor where the noggin and codin binds. The BMP is binding to this receptor, and so that noggin or codin could not able to bind, and that's why you do not have the formation of the nervous tissue. You have formation of epidermis. Or if you culture the animal cap, intact animal cap, without dissociating alone, it forms epidermis. Or if you culture the intact epidermis, animal cap, with this protein, either noggin, follistatin, or codin, it forms the nervous tissue. That means those are the signals, those are the stimulus which stimulates these cells to differentiate to nervous tissue. Or if you culture those things with excess BMP, so you have excess BMP, the bone morphogenetic protein, then it again it competitively bind to this it gives it compete with noggin receptor or codin receptor or follistatin receptor. So you have binding of more BMP, then you have formation of epidermis. So there is always a balance. So if the cell or if the system is producing more nervous tissue, you have generation of the BMP signal. Now the BMP signal will compete to this and bind to the noggin receptor. Then you have shutdown of this neural differentiation or formation of a neuron. Then you have shutdown of this signaling pathway. So you don't have formation of nervous tissue. So always in the body or in the cell, there is always a homeostasis, like how much noggin should be produced, how much codin should be produced, or follistatin or BMP. So this is just whatever I have told you. These are already written here, so you can go over this slide, what is required for formation of nervous tissue, or who excess BMP, how it inhibits the formation of nervous tissue. Everything I have just written in the slide, which I have mentioned. So if I summarize the story so far, we can show that we can say that the mesoderm, as I mentioned, it invaginates from the dorsal blastopore leaf during gastrulation. That induces, that forms the mesoderm and notochord that actually induces the formation of nervous tissue. There are specific signal called as noggin, codin, which are secreted by the dorsal blastopore leaf that forms actually the Spearman organizer that actually forms the nervous tissue. And the secretion of a noggin or the action of a noggin or codin is blocked by the secretion of the BMP molecule. So then the next slide I'll be talking about some of these studies, genetic studies people have done in the noggin mutants or codin mutant to say whether noggin, codin, they are sufficient to form all the nervous tissue, or there are some other signals which also induce formation of nervous tissue. Or just want to say how the anterior posterior, because you have the formation of anterior posterior axis during the development. You have anterior nervous system, you have posterior nervous system, like the hindbrain, midbrain. The hindbrain, those are generated during the anterior development of nervous system. Then what happens to the posterior part? So, or noggin or codin, they are sufficient for the development of all these anterior posterior axis. So I'll be talking about soon. There are some study people have done in the animals to say that noggin or caudin or polystatin, it is not all sufficient to develop all the nervous tissue. So this is a wild type mouse, and this is a noggin knockout mouse. So in the noggin knockout mouse, in the wild type mouse, you can see that you have formation of these nervous tissue, you have formation of ear, and those things are missing in the noggin knockout mice. But you have the formation of the dorsal nervous tissue even in the noggin knockout mice. So, or in the noggin or caudin double knockout mice. So how how this is happening? What are the other signal that is specifying the posterior nervous tissue? So those signals people have also identified. Those are called as planar signal, and I'll be talking about soon. What are the planar signal that specifies the posterior neural structure? So there are two. Actually, there are two-step model during anterior posterior patterning. One is activation model, another is the transformation model. So in activation model, what happens? When there is a formation of activated ectoderm, that actually develops into forebrain, the anterior portion of the nervous system. The anterior portion of the nervous system or forebrain is developed because of the action of noggin, caudin, and polystatin. But the posterior portion, the hindbrain or the spinal cord, they are developed through the action of planar signals. Those planar signals, these are the Wnt signal or retinoic acid and other signal I'm talking about soon. So you have first the activation and you formation of activated endoderm, and that actually forms the forebrain, the anterior portion of the animal, the nervous system, and during transformation, those signal actually those posterior signal that I'll be talking about soon, that forms into hindbrain and spinal cord. So those are here some of the experiment we have done in this. This is another development of the chick embryo. So here in the ectoderm you have forebrain, midbrain, and hindbrain, and in the mesoderm produces like different signal, like radial signal called as caudin, noggin, and polystatin, and those give signal to the formation of the forebrain or the anterior structure. And in the mesoderm you have another set of signal, which is the planar signal that form the posterior patterning, that form the posterior portion of the nervous system. Those signals are fibroblast growth factor, the Wnt signaling, or retinoic acid. So here one of the experiment people have done actually to say that the posterior patterning happens because of the balance of the production of the retinoic acid. So when you have low retinoic acid, it permits the anterior Hox gene to be expressed. So when you have low retinoic acid, you have formation of anterior axis. But when you have high retinoic acid, you have formation of posterior formation of the posterior axis. So the anterior axis and the posterior axis is defined by the accumulation of retinoic acid, and also by the action of radial signal like caudin, noggin, or polystatin. Caudin, noggin, and polystatin, they form the anterior axis, and the retinoic acid or the Wnt signal or the fibroblast growth factor signal, they form the posterior axis. So there is action of dual action of either the radial signal that called as the caudin, noggin, or polystatin, or the posterior signal that is the planar signal that is called as fibroblast growth factor, Wnt, or retinoic acid. So here is the experiment, the patterning of the posterior axis. So when they have incubated the fibroblast growth factor in the chick embryo, you can see that you have formation of the posterior axis, you don't have formation of anterior axis. And this is the control, that you have formation of anterior as well as posterior. When they block the fibroblast growth factor signal, you can see that the posterior axis is not developed. So anterior posterior patterning, the take home message is the anterior posterior patterning is defined by the action of two distinct signal. One is the planar signal and then the radial signal. The radial signals are caudin, noggin, and polystatin, and the planar signal are Wnt fibroblast growth factor or retinoic acids. So they, by the action of these two signals, they define the nervous system. They generate the nervous system in the animal. So this can be summarized in the last slide. So here this is whatever I told you earlier, like everything is summarized here. So this is the example of amphibian embryo. So you have formation of dorsal blastopore lip, and you have invagination and formation of mesoderm. So here two distinct kind of signal that defines the anterior posterior patterning of the nervous system. One is the generation of the radial signal. These are the white arrow. You can see these are the radial signal. So at the edge of leading edge of the ectoderm, endoderm and mesoderm, you have a generation of this radial signal that define the anterior that form the anterior axis of the animal, and which further develop into forebrain. And by the action of this planar signal, the Wnt or the fibroblast growth factor or retinoic acid, they forms this, they transform this animal to the posterior, they develop the posterior portion of the nervous systems. So the take-home message is, those are the signals that induce nervous tissue, the radial signal and the planar signal. So just remember that the radial signals are, the noggin, cordin, or polystatin, or the planar signal is either retinoic acid or fibroblast growth factor or Wnt signals. So by the action of these two signals, you have formation of the induction of the nervous tissue, either anterior axis or posterior axis. And the BMP always you have, when you have excess formation of this nervous tissue, so BMP always inhibit or kind of homeostasis. It inhibit the formation of excess nervous tissue, so that there is always a homeostasis or balance maintained in the system. So any questions? So this is the end of the slide. So if you have any question, we can discuss more because we have a lot of time. Any questions? Thank you.